Hello students, a warm welcome to all of you in ITLS Academy and also welcome to all in our 3 months online certification course on topic nanobiotechnology. In today's lecture, we will discuss about the receptor based biosensor. Okay, now we start today's lecture. What is a plasma membrane? Plasma membrane is a outer surface of cell. It is a outermost membrane of cell. It is present in both animals and animal and plant cells. It is made up of phospholipid. It is made up of phospholipid. Okay, in the bilayer form. Bilayer means it contains two layers. First layer and second layer. Okay, and this is a head part and this is a tail part. This is a hydrophilic part and this is a hydrophobic part. Okay, and the main function of plasma membrane is to generate or to generate a important sensory interface between the cells and extracellular environment. Okay, it will act as a sensory interface. Okay, this phospholipid bilayer contain a heterogeneous mixture of lipid and membrane bound proteins, and these membrane bound proteins are helpful for the transduction of molecules, for the transport of molecules across the plasma membrane to facilitate the recognition, transduction and amplification of extracellular signal. Biological sensing at the cell membrane start with the binding of extracellular ligand to its complementary receptor, and this receptor is present on this cell membrane. Okay, this is followed by the transduction of binding element into the cellular signaling cascade. Okay, receptor engagement may the result in the conformational changes in receptor proteins, maybe binding or dissociation of accessory proteins or phosphorylation of the receptor or downstream mediator. Whenever this ligand is bind to this receptor, the resulting some conformational changes occur into this receptor. These conformational changes may be binding or dissociation of accessories protein, phosphorylation of receptor and downstream mediator. Okay, after this, whenever the primary messenger will bind to the receptor, then these conformational changes will occur. After these conformational changes, secondary messenger will facilitated and intracellular signaling. Okay, intracellular signaling cascade that cumulates in the eventually cellular response. Okay, membrane receptor have evolved to sense a vast variety of endogenous and exogenous compound and are integral in a key cellular process ranging for the central nervous system, cardiac function to sensory systems such as smell and vision. Okay, now we understand the biosensor. What is a biosensor? Okay, biosensor combines the uses of biological sensor with the physical transduction machines, which transmit the information into the measuring signal. What is a biosensor? Biosensor is a type is the combination of biological sensors with physical transduction mechanism, okay, which transmit the information in the form of measuring signal, okay. Biosensors have been constructed using the biological components such as enzymes, antibody, nucleotides, pores, ion channels, and receptors, okay. Affinity based biosensor is a affinity based sensor which take advantages of binding of a specific ligand or analyte to the biological molecules, okay. This term biosensor is short for the biological sensor, okay. The device is made up of transducer and biological elements, and these biological elements may be enzyme, antibody, nucleotides, and etc. The bioelement interact with analyte being tested, and biological response is converted into the electric signal by the transducer. This transducer convert the biological signaling into the electrical signaling. Depending on their particular applications, biosensors are known as a immunosensor, resonant mirror, chemical canaries, biochips, glucometer, and biocomputers. Okay, what is a biosensor? Biosensor is a combination of biological sensors with the help of transduction, physical transduction. And what is the role of transduction? Transduction will convert the biological sensor into the electrical signal, okay. And by the help of this physical transduction machine, we transmit the information into the measurable signal. And these biological systems, these biosensors has been constructed with the help of biological component, for example enzymes, antibodies, nucleotides, receptor, ion channels, etc. Now we understand the working of biosensor with the help of this diagram, okay. This is a analyte, and here this analyte act as a ligand, and this is the biological receptor, and these bioreceptor may be enzymes, may be antibodies, okay. Whenever this ligand bind to this receptor, then some conformational changes will occur in this receptor, and these conformational changes is recognized by the help of this transducer, and this transducer will convert the biological system into the biological signal into the electrical signal. Then this transducer will further proceed this signal into this amplifier, and this amplifier will amplify these signals, okay. Then these signals are processes by the processor, and then finally display on the result in the form of result. This is a basic working of any types of biological sensor or biosensor. First we require an analyte, then receptor, then transducer, then amplifier, then processor, then finally display. Analyte is act as a ligand, and this ligand will bind to this receptor, and this receptor, there are a certain types of conformational changes will occur in this receptor, and these signaling and these changes are detected by the help of this transducer, and this transducer will convert the biological system into the electrical signal, and then further this signal is transferred to this amplifier, and this amplifier will amplify these signals, and then processor will process this whole process and finally display on the computer screen and any other instrument, and this is finally result, and this is the finally working of biosensor, okay. Now we understand the olfactory G protein coupled receptor or GPCR signaling. This olfactory receptor belongs to the G protein coupled receptor. Okay, GPCR is involved in numerous signaling recognition and transduction events, that is the sensing of hormones, neurotransmitter, ions, proteins, and many other events also. Since GPCR are involved in many path, path pathophysiological process, it is not surprising that more than 50% of pharmaceutical on the current market target this receptor for the for the drug for the hmm. Since GPCR are involved in many pathophysiological processes, it is not surprising that over the 50% of pharmaceuticals on current market target this receptor. Okay, what make GPCR ideal for the biosensor technology in their sensitivity and affinity for wide range of chemical ligands. In additionally, the field can take advantages of breadth of knowledge we have acquired over recent decades into the structural functional properties of these proteins. Okay, the majority of interest in GPCR sensor developed has focused on their uses for drug discovery. These assays take advantage for measuring the intercellular 3' to 3' to 5' secondary messengers such as calcium and cyclic adenophrine monophosphate, and maybe use the some fluorescent level dye to target these downstream signaling molecules. Okay. Now we understand the GPCR receptor or olfactory receptor. Okay, this is a GPCR receptor. GPCR receptor. And it is a type of olfactory receptor. Okay, this is a binding site for ligand. And this is the transmembrane proteins. Okay, these are the transmembrane proteins. And the number of transmembrane protein is 7. Okay, and this is a ligand or target molecule. Odorant. Okay, and here odorant is act as a odorant molecules is act as a ligand. Okay, this GPCR or G protein complex contain a heterotrimeric structure. It is a heterotrimeric structure. Heterotrimeric structure. It contains 3 sites. First alpha and second beta and gamma. This beta and gamma is always associated, and this alpha subunit is dissociated when the receptor and when the ligand is bind to this receptor. Okay, now, and this is a adenyl adenylate cyclase enzyme. It's a type of enzyme. Okay, now whenever these ligand or odorant is bind to this receptor, then GTP is converted into the GDP. This is a alpha subunit and this is a beta and this is a gamma. Okay, now this receptor or ligand is bind to this receptor and bind to this receptor, then here some conformational changes or some reactions are occur. In this process, the GTP is converted into the GDP. Whenever this GTP is converted into the GDP, then this alpha subunit will dissociated. Okay, whenever this alpha subunit is dissociated, it will transfer or start the intracellular signaling. It will start the intracellular signaling. Okay, and it will convert the ATP into cyclic AMP. AMP means cyclic adenosine monophosphate with the help of this adenylate cyclase enzyme. Okay, then this C cyclic AMP is act as a secondary messenger. It will further proceed the signaling and this calcium or potassium channel will open and this calcium and potassium will migrate towards the influx into the cell and this chloride channel will open. This is a chloride channel and this chloride channel will open. Whenever this chloride channel is open, then this chloride will influx or move outside the membrane. Whenever the calcium or potassium migrate inside the cell, then chloride and calcium channels will open and this chloride ion will move outside the membrane. Okay, now resulting due to this whole process, this cell membrane will depolarized. Okay, and generate the action potential or electrical signal. Okay, and this signal is transported to the olfactory bulb and cortex, and then further process will conduct. Okay, this is a whole processing of GPCR or olfactory GPCR receptor. In this process, first this ligand bind to this receptor, then GTP is converted into the GTP, alpha subunit will dissociated and this alpha subunit will dissociated and ATP is converted into the cyclic AMP with the help of this adenyl cyclase enzyme. Then the calcium and potassium channel will open and calcium and potassium will migrate towards the membrane. Then These calcium and potassium will open the calcium chloride channel, and these chloride is migrate outside the membrane, and resulting this process, this plasma membrane will depolarize and action potential is generated, or electrical signal is transmitted to the olfactory bulb and cortex. Okay, this is a plasma membrane. Okay, this is a whole working of olfactory receptor, G protein coupled receptor. Or okay, now we understand the olfactory receptor. Now we understand the olfactory receptor. These olfactory receptor comprise a large number of GPCR proteins. Okay, and these receptors belong to the rhodopsin or class A family of GPCR. This is likely due to the ability of higher primates that effectively coordinate their sense, such as vision or smell. These olfactory receptors are present on the olfactory sensor neuron, such as olfactory epithelium of the nose. A unique aspect of olfactory system is one of the olfactory neuron express are distinct types of olfactory receptor are concentrated on the cilia of the neural dendritic that faces the nasal lumen, where they come in the contact with the odorant or ligand either directly or after the odorant has been captured by the binding protein located in the olfactory mucosa. Upon OR ligand engagement, signaling is initiated as explained earlier. We already discussed the functioning or signaling of olfactory receptor in our previous slide. Okay. Culminating in transmission the electric signal down to the axon of the olfactory bulb and further into the olfactory cortex of the brain. Okay, whenever the odorant is bind to the receptor, then signaling is started. Whenever then after that signaling, this signal is transmitted to the axon of olfactory bulb and then transferred to the olfactory cortex of the brain. Okay, giving their physiological role of olfactory receptor are not presently target for any drug discovery. The unique ability to identify the largest variety of chemical with great precise makes their group of GPCR very attractive for the biosensor development. Now we understand the olfactory biosensor application. The electronic nose or e-nose was first introduced in 1982 by Pursuit and Dot. Okay, electronic nose are based on the interaction of volatile with various surface chemistry and consequently signaling being transduced to the signal to a sensor such as semiconductor, piezoelectric and optical sensors. Okay, this electronic nose was the electronic nose was first introduced in 1982 by Pursuit and Dot. Okay, the electronic nose are based on the interaction of various volatile substances and various surface chemistry and consequently signal is being transduced by the sensors, and these sensors may be semiconductor, piezoelectric or optical sensor. This electronic noise has a lots of application in real life application such as food and fragrance industries to smell the food and different types of fragrance. Okay, to detect the toxins, environment monitoring and medical diagnostic. A specific example include the biosensor for breathing analysis to detect alcohol, disease-related volatiles such as acetones, gas and exposure of sensing and detecting of volatile from food spoiling microorganisms. These are the application of electronic nose. Okay, while much progress has been made in electronic nose development, this technology is still lack of considerable sensitivity. Okay, we when compared the affinity of ORS to the natural ligands. Okay, our olfactory system is more sensitive than any physical method of chemical detection. Okay, whenever we compare this electron nose to their natural ligand system, then there are a lack of sensitivity. Okay, our natural olfactory system is more sensitive than any other physical method for chemical detection. Electronic nose has applied to sensing of chemical and volatiles in a number of real life applications. This electronic nose has a lots of this electronic nose has a lots of application in our real life. For example, food and fragrance industries, detection of toxic environment monitoring, medical diagnostic, okay, for breath analysis to detect alcohol, disease related volatile such as acetones, gas, and exposure of sensing and detection of volatile and food spoiling. ये वाली नहीं वही अच्छी थी. ओके, लास्ट वाली स्लाइड्स, जो वाला अच्छा वाला कंटेंट है उसी में से लेना है, पर जो अब वाला कंटेंट है वो इसमें से लेना है. कैसा? While much progress While much progress has been made in electronic nose development, this technology is still lack considerable sensitivity. Whenever we compare this technology to affinity of natural olfactory receptor, then there are some while much progress has been made in electronic nose development, but there are some lack of sensitivity. Whenever we compare this electronic nose with the natural olfactory ligand system, okay, our olfactory system is more sensitive than any other physical method for detection of any chemicals. Okay, the natural progress the nat okay, this is a application. Okay, now this is the application of olfactory. This is the application of olfactory receptor based biosensor. This is the application of olfactory receptor biosensors. This is the application of olfactory receptor biosensor. Okay, now we understand the immobilization of receptor onto the biosensor surface. Now we discuss the immobilization of recombinant onto the biosensor surface. Firstly, we discuss the expression and purification of recombinant PCRs. Okay, this PCR used in a cell-free biosensor and require an adequate expression of protein isolation and purification system. Okay, this is a very big challenge in the field of GPCR or olfactory receptor, as many of these receptors are difficult to express in large quantity as recombinant protein. Okay, for the isolation of recombinant proteins, researchers use the mammalian or insect system for proper protein folding and processing. Okay, rather than any mammalian system. Okay, however, due to the relative as and low cost of the prokaryotic system, prokaryotic expression system, some effort has been made to enhance the solubility of recombinant receptor derived from this method. Which method? Prokaryotic expression method. In prokaryotic system, basically we use the E. coli expression. The eukaryotic expression system is too much costly and time costly as compared to prokaryotic expression system. Okay. Recent study has been investigated the use of cell-free product of olfactory such as the use of wheat germ extract. While mammalian systems are often preferred use of insect cells such as the baculovirus used in the SF cell line is feasible alteration of mammalian expression system as these cells maintain the adequate post-translation protein processing and membrane incorporation. Okay, the other advantage of SF cell line over the mammalian cell line is that these insect cells are largely lack of endogenous G protein signaling component, which can interface in the measurement of recombinant protein function. Okay, further, insect expression system achieve large protein yield and relatively easy and cost effective or inexpensive to culture compared to the mammalian system. This is the isolation process or isolation expression system of recombinant protein. And for the purification system, we use the lots of purification method such as dialysis, chromatography, etc. Now we discuss the immobilization of lipid bilayer and membrane-bound protein on the sensor surface. The difficulty in using the membrane protein for biosensor application is needed for the hydrophobic nature. Why we use this hydrophobic nature? This hydrophobic nature of lipid bilayer is required for the correct structure protein conformational and their function. Okay, for the therefore most of the cell-free biosensor application involve the GPCR utilize a supported lipid bilayer. The lipid bilayer is synthesized from the phospholipid, applied the physical transducer and protein of choice inserted into the membrane. Okay. This assembly ensures a consistent distribution, known protein concentration and lack of interference from other membrane protein found in membrane fraction of the cellular expression system. This assembly ensures a consistent distribution of known protein concentration and lack of interference from other membrane protein found in membrane fraction of the cellular expression system. Okay, for the immobilization of membrane bound protein into the sensor surface, we need the hydrophobic nature. Why we need the hydrophobic nature? Because due to this hydrophobic nature, proteins will maintain their structure and their conformational structure and functions. Olfactory receptors are difficult to reconstruct into the artificial lipid system. Okay, it is very difficult to reconstruct the olfactory receptor into the artificial lipid membrane, or mostly study have therefore relied on immobilizing crude and purification membrane fraction from the heterologous expression system containing the recombinantly overexpressed proteins. Okay, this is largely prevent the issue of integrating the receptor protein into the artificial lipid bilayer in the correct orientation. Okay, for the immobilization of lipid bilayer on the solid surface, basically we use the two types of method. Basal fusion method and second is the leguminar Boldent method. Okay, Both method take advantages of self-assembled nature of surfactant. Okay. Small vessels in solution rupture on the absorbed on the surface and fuse to the form of flat lipid bilayer. Okay, this lipid bilayer may be absorbed on the physical sensor by using self-assembly monolayer or by the deposition of cushion polymer shoe coat. Okay, eliminating the direct contact of the understanding the bilayer with the solid surface can help to maintain the structural integrity of membrane protein. Okay, for example, compared to the direct absorption method. Okay, in direct absorption method or direct absorption of cell membrane onto the solid sensor surface, membrane protein can directly couple with the solid surface by the help of membrane linker, streptavidin biotin link or antibody. Okay, antibody, and this increases the amount of receptor onto the surface and as well as adding the directionality of receptor placement. Okay, now we understand that, and now we understand this with the help of an example. A scientist Goldsmith and his colleague immobilized the recombinant mouse olfactory 174-9, mouse olfactory 2031, or mouse olfactory receptor 256-17 proteins containing a terminal histidine tag. Histidine tag is a type of membrane linker. Okay, to carbon nanotube. Histidine to carbon nanotube functionalized with nickel nitrilotric acidic acid. Now we understand the physical sensor platform. Unlike channel protein which transmit ions following conformational changes in response to ligand binding, GPCR do not have the electrochemical signature. Okay, and sensor therefore need to be sensitive enough to be able to detect the alteration event. Okay, the majority of example explained here depend on the receptor ligand binding induced protein changes. Here a number of techniques are described that have recently been employed on olfactory receptor biosensor application. Okay, these do not rely on the labeling technique for visualization of the binding event. Okay, now we understand these techniques. Label-free assays are attractive as they removed an additional component that might have stability, cost, and decrease the signal to noise ratio relative issues. Okay, the following technique will be discussed in the context of, here we discuss the technology which is based on the cell-free system. Okay, now however, it should be pointed out that these have been successfully applied to the whole olfactory neuron and tissues, serving as an alteration to the traditional electrophysiological technique such as patch clamp. Okay. For example, now we understand this technique with the help of an example. For example, now we understand this technique. Now we understand the technique of physical sensor platform. Okay, field effective transistor is a type of device. Okay, whenever we coupled the Colado potato antenna of Colado potato whittle with the help of field effective transistor, then researcher is able to detect a chemical, and this chemical is 3-hexane-1-ol, or this chemical is released by the plant whenever the plant is damaged by the insect bites. Okay, Now, olfactory response has also been measured in intact olfactory epithelium tissues, olfactory neuron or cells, expression, recombinant olfactory receptor with a variety of sensor device including the microelectrode array or quartz crystal microbalance. This is a physical sensor platform. Now, this is a type. Now we understand the field effective transistor is a type of physical sensor platform. Okay, this is a great interesting in using the micro and nano electronic device for biological sensor. These has been successfully applied to the whole cell olfactory receptor sensor studies. Technique take advantage to changes in electrical properties after surface has been functionalized with the molecule of insect. Okay, electronic chemical impedance spectroscopy has been used to test the affinity of number of odorant to the red or the olfactory 7 protein immobilized on the sensor surface protein in the membrane friction. Electrochemical impedance spectroscopy has been used to detect the affinity of the member of odorant to the red olfactory receptor 7 protein immobilized onto the sensor surface in the membrane friction. Okay, this technology is also used to detect this was shown to be sensitive detection are based on the modeling and experimental result. Okay, electrochemical impedance spectroscopy to detect the specific protein conformational changes following by the ligand binding based on the impedance characteristics. And this electron chemical impedance spectroscopy is used for the detection of protein conformational changes. Okay, silicon based field effector transistor is the combination with carbon nanotubes provide a sensitive platform that enables the size reduce the multiplexing of individual devices. Okay, functionalization of biological material onto the carbon nanotubes and field effector transistor affect the current voltage characterization of carbon nano. Now we understand this acoustic wave sensor. Okay, quartz crystal macrobalance and surface acoustic wave sensor platform take advantage of acoustic wave generation of piezoelectric material. Okay, basically the basic function of this sensor is based on the changes in resonance frequency of piezoelectric material, and these piezoelectric material may be quartz plate or following binding of molecule on the to the surface. Okay, it is believed that this technology not only sense the changes in mass but also detect the conformational changes of immobilized molecule. This quartz crystal microbalance first utilized in the combination of olfactory receptor whenever we extract the crude protein from the wolf fog olfactory receptor, then these extracted protein is absorbed on the quartz plate, that is a type of piezoelectric material, and then so the response of this protein against the different odorant, and then finally we observed our result. This is acoustic wave sensor, and this is all about today's lecture. Please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share and subscribe ITLS Academy. Thank you so much.